Have you ever wanted to shapeshift?
Wait, what? Turns out all you may need to do is just change your behavior and environment. My DNA doesn't change? Doesn't my DNA determine what I look like?
Well, yes. But before we go any further into figuring out how exactly our behavior and environment can change the way we look, let's take a look at the basics of how our DNA is expressed. Through the process of gene expression, our DNA is synthesized into proteins and other functional products that make up our phenotype, or also known as our physical being. In a cell's nucleus, single-strand RNA molecules read and copy chemical bases in DNA through transcription. Let's take a closer look. RNA polymerase first binds to the DNA sequence at the promoter, which is found at the beginning of the gene needed. The DNA strands are then separated and act as a template for an RNA transcript, containing the copied chemical bases to be formed. The RNA transcript is then released from the RNA polymerase once the terminator is reached. Then, through translation, amino acids are synthesized by ribosomes using RNA to create proteins and other functional products that make up your physical being. However, your behavior and environment can affect the way your genes work.
Also known as epigenetic changes, your genes can be turned on or off. One of the ways in which this occurs is through DNA methylation.
Or when a chemical group, a methyl group, is added to a specific place in DNA. It blocks the RNA that reads the DNA, and hence the gene is never transcribed. But epigenetic changes are reversible, so methyl groups can be removed through a process called demethylation. Another example of epigenetic changes is histone modification. Our DNA is wrapped around proteins called histones, forming chromatin. When methyl groups attach to histone tails, they enhance their positive charge, causing DNA to wrap more tightly around the histone, making it more difficult for RNA polymerase to read the DNA sequence. However, when other chemical groups, such as acetyl groups, attach to histone tails, the positive charge of histone tails is neutralized, and the epigenetic change is reversed. These changes can occur at any stage of gene expression, not just transcription. Unlike genetic changes, epigenetic changes only change how your body reads a DNA sequence.
So you may have genes that can code for all sorts of physical features, but it is the expression of your genes that determine whether they can be seen or not.
Unfortunately, we have yet to discover what exact behaviors or environments trigger epigenetic changes. However, there has been previous research into the field of epigenetics that can help us better understand this process. In 2004, Weaver et al. Investigated the influence of nurturing on a rat's vulnerability to stress. Laboratory-bred rats and their mothers were categorized by their mothers' licking and grooming behaviors. To begin with, hippocampal tissue was taken from all the offspring of the mothers. Genetic mapping showed that there were higher levels of methylation in the glucocorticoid receptor gene in the offspring of low LG mothers than in the offspring of high LG mothers. Another group of rats had the offspring of the mothers with low LG behaviors swapped with the offspring of high LG mothers. After genetic mapping, the rats who were raised by the low LG mothers showed the same patterns of methylation in their glucocorticoid receptor gene as the biological offspring of low LG rat mothers in the first study. Finally, the rats of the cross-adoption study were placed in a plexiglass restrainer, with blood samples taken before and after this procedure. The results showed that corticosterone, a stress hormone, was significantly more concentrated in the blood of those raised by low LG mothers, those whose glucocorticoid receptor gene was methylated. As the glucocorticoid receptor genes ensure the production of glucocorticoids which regulate a rat's response to stress, methylation of this gene would have meant that the rats would have produced more stress hormones than necessary. This showed that vulnerability to stress could be determined by the infant environment of rats. Although these results enable us to see and understand how our environments can affect our behavior and phenotype, the results of this particular study cannot be directly applied to human beings, as our DNA and physical structure is obviously not the same as a rat's. So shapeshifting at will isn't exactly possible at the moment. But understanding epigenetics can help us to understand aging, find ways to diagnose cancers, and may even lead to personalized medicine. But for now, I guess I'll just stay the way I am.